And welcome to our In Focus discussion tonight, CRISPR and genetic editing. People have been, quote, editing genes for a long time now, selective breeding of food plants to make them more productive, or animals like dogs or cattle to give them characteristics we like. But a technology called CRISPR has made that process easier than ever. So what does that mean for us as individuals and as a society? Joining us now in studio is James Tabery, professor of philosophy at the University of Utah, has been focusing on the ethics of modern day implementation of genetics. We'll call you Jim, right? That's what you prefer?
Jim it is.
All right. And also Dana Carroll, professor emeritus from the School of Medicine at the University of Utah and one of the founding fathers of genome editing. Thank you both for joining us here this evening.
Good to be with you, Emily.
All right, we are breaking this down for all of us who may not be nearly as smart as you are on this topic. So on a basic level, Dana, what is gene editing and how does it work?
Hi, Emily, and thanks for having me here. Gene or genome editing is what we call the capability we have these days to send an enzyme into cells and make a change at a very specific, very tiny little location within all of the DNA of all of the chromosomes inside the cell. And these changes can be disabling a gene, or they can be making a gene have a novel capability. And it's something that geneticists have wanted to be able to do for a long time. And for the last 15 years or so, we've had that capability.
Can you tell us, when we ask how does it work, just some of the things that have been most fascinating to you that you've seen that you're now capable of? What are some of those things? Oh, that's you still, Dana.
Sorry, how does it work? Well, we have these enzymes. We can program them so that when they get into the cell, they go to a very specific location. And you program it for one location, it'll go there, it'll make the change. You change your mind tomorrow about what you'd like to do. The enzyme now, with your instructions, goes to that other location, makes a change there. So if you're working on one gene today, another gene tomorrow, and a third gene on Thursday, you can modify this enzyme so that it attacks each of them when you want it to.
Absolutely fascinating. Okay, Jim, people have been doing editing genes for a long time with breeding. Talk about that as a starting point.
Yeah, exactly, Emily. You know, look no further than the nearest bulldog to see what humans are capable of doing to the organisms around them. Humans have been altering the genomes of livestock and crops for millennia, ever since humans migrated to an agricultural way of life. Trying to breed cows to create more milk, or breed wheat so that it provides more yield of grain. The big difference between that and what we're talking about tonight, at least one of the big differences, is time. What we saw with selective breeding of cows and pigs, grains, played out over decades or centuries or even millennia. With the gene editing technology, the potential is there to do that in just one generation. And on the one hand, that opens real opportunities that people are excited about. You see people talking about just one generation breeding sheep that have more muscle mass, which means more meat to sell, or breeding racehorses that have more muscle mass so that they can run faster. On the other hand, one thing that we need to be mindful of is these sudden changes. The organisms aren't necessarily ready for that. You could build a faster horse or a more muscular sheep, but if its ligaments, its tendons, if its skeletal system isn't actually equipped to handle that, then you can have a real problem on your hands and you could actually induce some suffering.
There is so much to this. You just think, yeah, put some more muscle on it or make it faster, but you're right, all of the snowball effect after that, what does that mean? Interesting. Well, Dana, let's go back to you. Can we define a couple of things right now? First, can you distinguish the difference between somatic and germline treatments? You're gonna have to definitely break those down.
Sure. So somatic genome editing refers to doing something in an organism that's already alive. So if we're talking about medical treatments, this would be having a treatment for an adult or a child that has a genetic condition that we might want to address. Germline editing means that you're going to do something that can be passed on to the next generation of that individual. So you have to do something that affects the sperm or the eggs of the individual that you're treating.
Okay, fascinating. Can you explain some different levels of complexity of treatment? How is, say, sickle cell anemia and Tay tactics different from something like congenital mental disorders? Again, you're going to have to break this down for all of us here at home.
Well, both sickle cell disease and Tay-Sachs disease are the result of mutations in a single gene for each of those conditions. And so genetically, we know exactly where those mutations are. We know how to address them. We know what changes to make so that they won't have the disease impact. With other sorts of diseases or other sorts of traits that people have, it's more complicated. There are multiple genes that contribute to it. So if you think of something like height, so Jim's children are probably going to be taller than my children because he's taller than I am. We know that height is at least partially inherited, but there are multiple genes that contribute. So making a change in a single gene will only have a very small effect on how tall someone will be.
This is absolutely fascinating to think how much you can do, but I think also that's why we have to have the discussion of the ethics behind it all. Dr. Carroll, Dr. Tabery, we'll be right back to talk more about the ethics of this technology. That's right after the break. You're going to want to stay with us. All right, thanks for staying with us for our second In Focus discussion tonight on CRISPR and genetic editing. We're here to continue our discussion with Dr. Dana Carroll and Dr. James Tabery, Jim, about the science and ethics of altering genes. So Dana, we'll start again here with you. How close are we to treating sickle cell?
There are already patients that have been treated for sickle cell disease using CRISPR. CRISPR is now the favored genome editing platform. It made things a lot easier. And so it's kind of taken over the field. But there was a paper published very recently showing that one sickle cell patient and one patient with a related hematologic disease called beta thalassemia had essentially been cured in the short term of their diseases using a CRISPR treatment.
Fascinating. Okay, and then Dana, continuing, let's just talk more complex things like height, muscle mass. Are those things that can be turned on by CRISPR technology?
Yeah, the one way we know to make greater muscle mass is to actually knock out a gene, not to turn one on. There's a gene that limits how much our muscles grow, and so we can use CRISPR or one of the other editing platforms to disable that gene. And as height, as I mentioned a moment ago, there are multiple genes that are involved, and so our ability to really control height is quite limited.
You, of course, have an in-depth look, both of you gentlemen, on all of this. So for us here at home, we just think, yeah, we talked about this when we were off camera and during the break, but turn on all the good things, turn off all the bad things. It's just as easy as that, but it's not. So even on a higher level of complexity, Dana, we'll just ask one more question before we head over to you, Jim. Can this technology boost even more complex things like intelligence, maybe through memory enhancement? I wish.
Well, we're really far from knowing the genetic influences on intelligence. And people are working on this, but I think it's, at least for the next couple of decades, this is a losing battle, just to figure out what it is in our genes that determines what our intelligence is going to be, even if you could define intelligence. So that's really hopeless to be able to use genome editing to modify. I think we're really not going to be able to do this in the short run. And even if we could make some of the changes that we might think would have an influence on intelligence, what other aspects of our makeup would those changes influence? The functions of genes are very intertwined within our bodies in different places in our bodies, and changing one will have an impact on many others.
I think that's what's interesting that I didn't necessarily think of, but again, another thing we were chatting about in the first block was, Jim, you said this, that if you add more muscle to the sheep, are their bone structures ready for that? So Jim, tell us, what do you think about this? How close are we to these potential changes in our own genetics, and do these changes worry you?
There are aspects of gene editing that I think should be recognized as ethically worrisome, but the thought that we're going to be altering babies' intelligence or their personality with this technology, that's not one of them. One of the things that came out of the Human Genome Project, we're coming up on its 20-year anniversary now, is that for most complex traits, when we think of diseases like heart disease, cancers, asthmas, or more personality traits like intelligence, cognitive ability, whether or not somebody's outgoing or introverted, those are impacted by dozens and even hundreds of regions in the genome. And then relatedly, many genes impact a number of different traits at the same time. And so because of that complexity, it makes it very difficult to tinker with that system. When you're dealing with rare single gene disorders, like Dr. Carroll mentioned, Tay-Sachs, sickle cell, cystic fibrosis, you've got one gene that you can target and work on. But when you're trying to deal with these more complex traits that are influenced by, in many ways, we don't know how many regions of the genome, you might find yourself, intervening in one place that has no effect on the thing that you were trying to impact and has some downstream effect on something else that you completely, was unexpected. And so that is a worry.
Yeah, so tell us, that's the thing. If that's a worry, what do you do about that? Do we just kind of put a halt for how far we go, or things that we should all be concerned about as a public?
Well, Dr. Carroll is a member of an international committee that is actually in the process right now of thinking through how to roll out this technology in a safe and responsible way. And so, I think it's going to be a process, and it's going to be a matter of moving from plant and animal models to human models, starting with embryos and moving on from there, starting with very simple genetic diseases and seeing what it's capable of, so that you're not sort of pressing these boundaries before we're actually ready to go there.
So fascinating. Okay, Dana, to really do some of this stuff, it has to be done at a very early time in a person's life. This is germline editing. Maybe during an in vitro fertilization. When in your mind would it be good to do this kind of gene editing?
You bring up an important point. Things like sickle cell disease can be treated in a child or an adult. We can take cells from the body, modify them with genome editing in the laboratory, and put them back into the same patient after the beneficial modification has been made. But for many things, we don't have a way to deliver the editing tools to the body, to the right cells, the right tissues in the body. Muscle would be an example. How many muscles do you want to affect? How are you going to get your changes to all those different muscles? So this can be done when an embryo consists of just a single cell, just at or just after fertilization. And a change that's made in an embryo will be passed on to every cell in the body all through development, into birth, childhood, and adulthood. And so that change will be one that will affect all tissues. Right. It will also be passed on to the next generation. And all of those aspects make me quite nervous about doing it with the technology that we have today. The technology is really not ready for that. And so the commission that Jim referred to, that I was on, set standards for what degree of competence you would have to achieve with this technology before it would be sensible to attempt doing things in embryo in conjunction with in vitro fertilization, even for the most straightforward diseases.
Fascinating to think all that you can do, but also being very aware of the standards that you should be abiding by, the responsibility at your hands. We have so much more to talk about. Stay right there. When we come back, we'll continue our conversation on CRISPR and genetic editing, again with Dr. Dana Carroll and Dr. James Tabery. We'll be right back. And welcome back to our third and final In Focus discussion tonight on CRISPR and the ethics of genetic editing. I'll tell you what, this has gone far too quickly for me. I hope those at home are still as entertained. We're going to pick up right where we left off with Dr. Dana Carroll and Dr. James Tabery. Jim, so changing the embryo. Let's talk to you, Jim. Isn't this eugenics?
Good question, Emily. A lot depends on where the technology is targeted. The eugenics movement, which was very popular at the beginning of the 20th century across the United States and even here in Utah, took it to be the case that biology or genetics could tell us what's the difference between a good birth and a bad birth. And eugenicists advocated for all sorts of measures to try to encourage more good births and try to discourage bad births. One of the sort of most frightening examples of that, which took place in the United States, was non-voluntary sterilization. Tens of thousands of people across the United States were sterilized against their will because they were deemed unfit to have children. The thought was that we don't want to have people like you. And in fact, here in Utah, hundreds of people were sterilized against their will for eugenic purposes. One of the things that was so frightening and wrong about eugenics is the traits that they were deeming to be bad births, in many cases were not genetic in nature, and in many cases they were just different ways of being. You know, they were disabilities, they were developmental delays. So, you know, I think as long as the gene editing technology is focused on traits that we think are sort of universally agreed upon as a bad thing, right? No parent would wish Tay-Sachs on their child, or sickle cell disease on their child, or cystic fibrosis on their child. When we're focused there, I don't think there's going to be much concern about eugenics. But there has been talk about shifting it to other traits. So an example of this is deafness. There's a genetic form of deafness, and there's been talk about using gene editing technology to eliminate this form of deafness. Many members of the deaf community don't think there's anything wrong with them, don't think there's anything wrong with their deaf children. They're proud of their deaf culture. They do not want to see American Sign Language go away. And so if we saw this gene editing move out of these straightforward, universally recognized problematic diseases to something that's more like just a different way of living, then I do think the concerns about eugenics are genuine and legitimate.
Absolutely fascinating. And sticking with you, Jim, this is also about designer babies, which is an economic justice question, right? Do you worry that wealthy people during, say, fertility exams could be taken into a room and given a list of options of how they want their kids to be, how tall, how smart? What about skin color, sexual orientation? This gets complicated.
It is. You know, I think if parents want designer babies, gene editing is probably the least efficient and most expensive way you could go about it. If parents want designer babies, they should give them good food, good education. They should raise them in space that's you know, devoid of environmental toxins. They should love them. Decades of research has made it clear that when we talk about the determinants of health, the things that make people sick or healthy, by and large, those are things in our physical, our chemical, our social environments. But you phrase it as an economic justice question, which I think is a really important thing to keep in mind. We already live in a world where wealthy parents are able to do things for their children that less well-off parents aren't able to do, right? They're able to give them better education. They're able to give them a healthier diet. They're able to raise them in neighborhoods that are not so close to toxic pollutants. And so that sort of, you know, we live in a world with disparity everywhere. Gene editing technology isn't going to create that, although it does have the potential to exacerbate it.
Fascinating. Okay. Well, Dana, let's talk about diversity in humans, especially from a genetic standpoint. What are the consequences of editing out diversity, even if it makes us healthier, so to say?
As Jim says, it'd be nice to edit out some of the diversity and the diseases that people suffer from. But if we really edited out large-scale diversity, that would be bad from both an aesthetic point of view. You know, diversity is great, having different people doing different things. But it would also be bad from a societal point of view because we want people with different types of talent and prowess, artistic talent, athletic prowess, intellectual prowess. And we also don't know what challenges the world is going to throw at us in the future. And if we have people with lots of different capabilities, sizes, shapes, and talents, then we're prepared for a lot of different things that may occur.
Yes. Okay. Well, Dana, let's stick with you for a moment. China tried to use CRISPR, HIV in human embryos in 2018 to make a human that was resistant to HIV. When should we as a society start having discussions about the ethics of this technology? And are there policies in place now for this technology?
Well, the discussion should start yesterday, Emily. This is something that lots of people have called for, but very few of us know exactly how to go about it. This program is one way to inform people about what's out there, what's going on, things that we should all be thinking about. There isn't any specific regulation in most places around the world on this technology per se, but since it's a medical technology and the uses we've been talking about, there are rules, regulations, laws about the practice of medicine that do cover a lot of the sorts of things we've been talking about.
Absolutely fascinating. Okay, we don't have much time left, but your final thoughts, Jim. What do you want to leave our viewers with tonight?
Well, the one thing that we haven't talked about, what I think is worth mentioning, is just access and cost. From what we know about how this technology works, the prices that are being talked about are in the hundreds of thousands or even millions of dollars. And so for most people, telling them that you've got a cure for their child, but it's going to cost that much money, is like telling them you've got a cure for their child, but it's on Jupiter. And so this is a fascinating technology. It's breaking science, but people need to be able to access it for it to actually do some good.
I think too, I know we're out of time, but one thing that you made mention of that hit me really hard is sometimes you think of these model babies, but really, like you said, your environment matters. The love in the home matters, the diet, what you're exposed to, all of that. We have a little bit of control, but outside of the genetic.
Absolutely.
Okay, well, we are absolutely so appreciative of your time. Again, for those at home, you've been hearing from Dr. Dana Carroll, Professor Emeritus of Bioengineering at the School of Medicine at the University of Utah, and Dr. James Tabery, a bioethicist at the University of Utah. Thank you both for helping us understand this complex, as we've learned, topic. Thanks again.
Thanks for having us.
Good to be with you.